Hi, everyone. It's a great pleasure to be with you today to discuss a story on unlocking the secret lives of proteins in health and disease. My name is Greka, and I'm an associate professor at Harvard Medical School and Brigham and Women's Hospital. So we live in an unprecedented time. It took several years and several $billion to sequence the first human genome, but now we can sequence an entire human genome in just a matter of minutes and with a fraction of that cost. So there's absolutely no problem in obtaining massive amounts of information about the genetics and genomics of human diseases, but we are now faced with a new challenge. That challenge is to actually understand how to decipher the meaning of all these genetics and genomics information to translate that into mechanisms of disease so that we can ultimately build targeted therapies, which of course means finding the right treatment for the right patient given at the right time. So how can we address this challenge of translating genetics into treatments? Well, to successfully execute on this arc of discovery from genes to mechanisms to medicines, I would like to suggest that it's now all about the proteins. So as we know, mutations in genes, of course, result in the wrong DNA, to be put simply. And that wrong DNA ultimately results in a wrong protein function. That ultimately leads to disease pathways because of the faulty protein function and perhaps the metabolites that are generated as a result of that faulty function. And so ultimately what we need to do is understand all these proteins and the functions that they have or dysfunctions that they have that cause disease so that we can ultimately translate them into targeted therapies. And I believe that this is true no matter what the modality, whether you're building a small molecule, a biologic, or even a gene therapy. At the end of the day, what we need to understand is the protein dysfunction that leads to disease. If you will, proteins are the business end of what we need to worry about inside cells so that we can ultimately build targeted therapies. So with this idea in mind, what I would like to share with you today is a story about the secret life of certain proteins that cause toxic proteinopathies. It's a story that started in my lab in the kidney, but as you will quickly see, it has applications far beyond the kidney in many different tissues and organs, and therefore gives us the opportunity to address many different diseases across the board. The story really begins about 20 years ago. A group of families on the island of Cyprus was found to have what seemed to be an autosomal dominant rare kidney disease that affected every single generation in those families. The geneticists were quickly able to identify a single haplotype that was responsible for this disease. But then for the following 2 decades, despite many, many efforts, it was impossible to identify the causal gene. It ultimately took the firepower of the Broad Institute and premier geneticists like Eric Lander and Mark Daly before we could ultimately identify the mutation in a dark little corner of the human genome, in a gene called mucin-1 and the variable tandem repeat region of that gene. My lab then took over to try to understand what that mutation means. It's a frameshift mutation that ultimately causes a toxic neoprotein, a protein that accumulates inside cells in the kidney and ultimately leads to kidney failure. As you can see in this knock-in mouse model shown in this picture, the mutant frameshifted protein is in green, and it accumulates massively in the areas of the kidney where the disease occurs in the tubules, leading to kidney failure in mice and of course also in humans. Armed with this understanding, we of course wanted to identify a way by which we could delete this misfolded protein. We were looking for a small molecule, and this took several years and many, many screens, but we ultimately identified BRD4780, a small molecule that, as you can see, is able to selectively clear out the green, toxic, misfolded protein while leaving the wild-type normal protein intact. We were very pleased by this discovery, but of course we wanted to see if it has the opportunity to work beyond the in vitro system, beyond the cells grown in the dish. And so we tested this in the knock-in mouse model that I showed you earlier. And as you can see here in the middle panel, there is a lot of green toxic protein accumulating in the kidneys of these mice. But after just 1 week of treatment with this BRD4780 molecule, we were able to effectively remove a lot of this misfolded protein from the mouse kidneys. What we were also very encouraged about was the fact that the small molecule also appeared to work in mini kidneys in a dish, if you will, what we call human kidney organoids, as shown on the far right. These are mini kidneys in a dish that are generated from pluripotent stem cells that were obtained from patients who were generous enough to give us their blood. We took their erythroblasts. This is patients and their unaffected siblings as controls. We derived human kidney organoids out of these pluripotent stem cells, and we were ultimately able to generate these kidney organoids. And as you can see in the middle panel, there was a significant amount of the green toxic misfolded protein accumulating in the organoids of patients. However, after just 3 days of treatment with the BRD molecule, as you can also see in the bottom right corner, the BRD4780 molecule was very effective in removing the green toxic protein from these mini kidneys in a dish. So now we had a small molecule that was able to work in cells, in mice, as well as in the kidney organoids, giving us a lot of confidence that this was a strategy that might work also in the clinic. And of course, we have been busy since then working on this new biology that we uncovered, not just in the kidney, but beyond, looking at tissues and cell types in the brain, in the liver, in the lung, in the eye. We think that this strategy and this new biology we have uncovered can address several rare diseases with unmet medical need. Just in the kidney alone, we estimate that about 150,000 patients in the United States can be helped by this approach, many more across the world. And taken together, if this strategy is applicable to all these other cell types and tissues and all these other toxic proteinopathies, we estimate that at least 5 million rare disease patients in the United States and many more around the world may benefit from this strategy. So what I shared with you today was a story of going through this arc of discovery from genes to mechanisms to medicines, essentially starting with mutations that appear to cause protein misfolding, deciphering that these toxic proteins accumulate in cells to cause disease, and using that information to ultimately build at least a starting point or an idea for a mechanism-based targeted therapy, hoping to take this discovery from the lab into the clinic. And of course, this is where we are now. We are very excited to be advancing preclinical studies in several animal models, not just in the kidney, but also in the eye and several other tissues. And this project is now ripe to transition from academic discovery to new co-formation. My team is, of course, very excited to work with a team of industry experts and pioneers to bring these treatments to patients. And we are very excited to share this story with you today as well. It's been a pleasure sharing all these ideas with you, and I look forward to meeting many of you and answering your questions. Thank you so much.